Hello, my name is Eric Procko. I am an assistant professor of biochemistry at the University of Illinois in Urbana-Champaign, and in my group we are interested in relating sequence to structure and function and the engineering of membrane proteins that are expressed in human cells. The approach we take to doing deep mutational scanning is to encode a library of different protein variants on an episomal plasmid that is then transfected using a large excess of carrier DNA to dilute down the coding plasmid into human tissue culture. And under those conditions, we typically get no more than one coding variant expressed per cell. So that gives us our tight link between a single genotype to a single phenotype. Unfortunately, under those conditions, 95% of the cell population will be negative for the protein of interest. So a lot of time is wasted on, for example, a cell sorter sampling negative cells, requiring that sometimes the FACS sorts need to be up to 10 hours in length over multiple days. But the advantage is that very quickly we can go from making a library to doing a selection within several days. So here's the basics here. We transfect our library, getting this link between genotype and phenotype in human cells, which then expressing our variants of our different membrane proteins. We will then add a fluorescent ligand, fluorescent substrate, maybe bimolecular fluorescence complementation, some kind of fluorescence-based method by which we can then use FACS sorting to pull out the cells expressing the protein variants of interest. And then we deep sequence and find out what's enriched and depleted. So I have three short stories I'm going to talk about. The first is on determining the structure of the sweet taste receptor. This is a class C G protein coupled receptor, or GPCR. This class of GPCRs is very interesting. They bind to a ligand in these large extracellular ligand binding domains, which are a long way away from the G protein coupling site in the cytoplasm for signaling. So ligand binding in some way triggers conformational changes in this obligate dimeric architecture. And so to understand that signaling mechanism, you'd like to know what it looks like. And when we started this project, at the time, only crystal structures had been solved for the isolated domains. So the ligand binding domain, the spacer domain, and the transmembrane domains. But how these domains came together in a heterodimeric or homodimeric architecture was unknown. So we were working on the sweet taste receptor. It's a heterodimer of two subunits, T1R2 and T1R3. When you express T1R2 in human tissue culture, these are all flow cytometry data here. So in red is transfected cells, and you can see that the monomeric T1R2 subunit can go to the plasma membrane for surface detection. However, T1R3, shown here in blue compared to the histogram trace of negative cells in gray, T1R3 can't go to the plasma membrane unless in green it is co-expressed with its heterodimeric partner, T1R2. So we thought, aha, here is the basis for an interesting selection to get its structure. We made a library on T1R2, and we sorted that library by FACS for folding and surface trafficking of the T1R2 subunit. And then we could do a second selection on the same library where we now looked for co-trafficking of the heterodimer with its partner. So the selection of monomeric T1R2 is shown at the top here, and then at the bottom is the mutational landscape for the selection of heterodimer trafficking to the plasma membrane. The protein's around 850 amino acids. It was split into three libraries, so around 17,000 mutations here. So on the horizontal, we are screening through the primary structure, the amino acid sequence, and on the vertical are the different amino acid substitutions. So orange means that a mutation is deleterious and was depleted following the FACS-based selection. And we were most interested in regions like this, where the mutations have very little effect on the T1R2 subunit by itself, but suddenly they are orange and deleterious for co-trafficking of T1R2 and T1R3 together. And we could map those regions to structure. So on the left is a crystal structure for extracellular domains, where we're looking at the mutational data, the conservation scores for the single subunit by itself. And then on the right, we're now looking at what changes, what becomes important and conserved, shown in orange, for co-trafficking of the two subunits together. So we see that there are these sites in deep orange. Here's site 1, site 2, site 3, and then a fourth site on the transmembrane domain. These are sites important specifically for heterodimer formation and co-trafficking. So now the problem is reduced to a domain docking problem. In the beginning, back then, we did this manually in molecular visualization software, but now we have, with Diwakar Shukla's team, we have a molecular dynamics framework where the adaptive sampling in the MD is guided by the mutational data to try to bury these conserved regions without changing the underlying energy function. So here was the model that we came up with for the dimeric sweet taste receptor. It's a very interesting model. It was immediately pleasing for a number of reasons. The main take home was that we could have this continuous folded structure here that enables coupling between the ligand binding domains and the transmembrane domains for signaling. So we could determine a mechanism for conformational change and for signaling. And we published our work almost simultaneously with a low resolution cryo-EM structure of a glutamate neurotransmitter receptor solved by the Nobel Prize winning group of Brian Kobilka. And our structures for the active conformation are virtually superimposable, and we arrived at very similar mechanisms. So I view this as a tremendous success for using deep mutagenesis to come up with unknown structures here for an entire family of important neuronal GPCRs where we could say something profound about mechanism. Unfortunately, we had a hell of a time getting this published. Editors just didn't think it was interesting. Who cares about this mechanism and this structure? But when it comes as a cryo-EM structure, then everybody thought it was seminal and amazing and brilliant. And I do fear that in this time period where now cryo-EM structures are becoming almost routine, if you don't have some tangible electron density, then the audience just doesn't care much. And that's a bitter pill to swallow, but it has meant that my group is really shifting towards engineering and away from determination of structures in dynamic membrane proteins. The second story I'm going to tell you about is involving a neurotransmitter transporter for serotonin. It's called SERT. It is the target of antidepressant drugs. And like many other transporters, the SERT transporter uses an alternating access mechanism. So these are slices through the structure here, where serotonin outside the cell can gain access to an outwards facing cavity, binds in a central substrate binding site to form an occluded conformation, and then an inwards facing cavity can open. Serotonin can then leave and enter the cytoplasm to complete the import process. With Diwakar Shukla, we have done extensive molecular dynamics of the import process. This conformational free energy landscape here is for SERT in the absence of a neurotransmitter. The deep energy wells are shown in blue, so we have an outwards facing conformation that can transition to an occluded state, but then shown in warm colors, there is an energy barrier to form the inwards facing conformation. And it's not until serotonin is present that now we have reduced energy barriers between these conformational states. So you have an energy accessible pathway to go from the occluded to the inwards facing conformation to release the serotonin to the inside of the cell. In my group, we were working with a fluorescent substrate called APP+. Here's the conformational free energy landscape. And what you'll notice is that APP+, it's a poor substrate, and it cannot reduce the energy barrier to form the inwards facing conformation. So there's this defect here. The conformational transition from outwards facing to occluded, that's no problem, but going from the occluded to the inwards facing state to complete the import process, that has this high energy barrier. We did deep mutagenesis on SERT for expression, shown at the top, and also for transport. The data quality is quite reasonable. So here in this top row, we're comparing log to enrichment ratio values for 2 independent replicate experiments. The agreement for these kinds of selections in human cells using our methods, this is quite good. We have some data sets that are much worse than this. We do have some that are also better. So again, orange means that mutations tend to be bad. So looking at the data for expression, here, this is a slice through the serotonin transporter structure. We find that these regions above and below what is the translocation pathway for the neurotransmitter, these regions are conserved, but the translocation pathway itself tolerates many mutations for surface expression and folding of the transporter, and that may be valuable for evolving new substrate specificities. When we now look at what changes for transport of the fluorescent APP+ substrate, then we find that the substrate binding site is now a very dark orange. It's highly conserved, but we find in dark blue many gain of function mutations that congregate and are enriched in this region, which happens to be the intracellular vestibule, so the exit pathway for the neurotransmitter. What mutations are we picking up for gain of function? Well, if we look at the hydropathy scores, yellow means hydrophobic residues are preferred, blue means hydrophilic residues are preferred, and we see that the intracellular cavity is now favoring hydrophilic polar substitutions. Why? Because these hydrophilic residues will now favor opening of the intracellular cavity such that it is exposed to solvent, exposed to water. So this is going to reduce that energy barrier from the occluded to the inwards facing conformation to favor the conformational transitions. This is showing it close up. Blue means that a mutation is a gain of function for APP+ transport. We get all of these blue mutations in the exit pathway. So in this region of the transporter, where again, they're trying to favor, through increasing hydrophilicity, favor the opening of the inwards facing cavity to enable substrate release into the cytoplasm. The third story I'm going to tell you about is engineering of receptors as soluble decoys for viruses. We work on a number of viruses in the lab, but I'm only going to show data for SARS coronavirus 2, the causative agent of COVID-19. So the SARS coronavirus has spike proteins on its envelope, shown here in bluish green, and these spike proteins engage through a domain known as a receptor binding domain, a receptor shown in red called ACE2 on a host cell. And this binding event then triggers fusion of the host cell and membrane and the viral envelope and infection. You can take soluble ACE2, where you chop off the transmembrane region, and it acts as a good neutralizing agent. It can bind to viral spikes and block infection. The problem is that the affinity of ACE2 and the viral spike is some tens of nanomolar dissociation constant. It's only moderate. You compare that to monoclonal antibodies for neutralization. They have affinity constants that are picomolar. So the problem we set out to solve is to engineer ACE2 to make it high affinity, comparable to a monoclonal antibody, to use as a therapeutic agent. The selection strategy was to make a library of human ACE2 expressed in human cells. We can detect the surface expression of variants in the library using fluorescent antibodies shown in red here, detecting an epitope tag. And then we looked at binding to the viral spike using the receptor binding domain, or RBD, again, fluorescently labeled. Here's what it looked like on the FACS instrument. The horizontal is the expression level of ACE2 in the cells. On the vertical axis is the degree of binding to the viral spike. So we collected cells in this green gate, which are expressing ACE2 variants that have elevated affinity for the viral spike. After deep sequencing, here's the quality of the data. So this is the agreement between two independent replicates. The nonsense mutations are shown in red. They're all deleterious. The non-synonymous mutations are shown in black. The experimental agreement between independent replicates is excellent. This is one of the best data sets we have in my lab. And so then we took several of the highly enriched mutations for binding to the viral spike, and we started combining them. This is the mutational landscape. Orange means the mutation is deleterious. So in the top row, all of the nonsense mutations, as you would hope for, are bad. But our attention was drawn to these blue mutations here. These are mutations that enhance affinity for the viral spike. So we combined 3 of them together to make a high affinity engineered ACE2 decoy receptor. Looking at the wild type receptor here, looking at biolayer interferometry kinetics, the first 2 minutes are looking at association, monovalent affinity here between wild type ACE2 and the viral spike. So there's a good association, but then after 2 minutes, there's also pretty good dissociation as well. And that limits the affinity to a KD of around 20 nanomolar. But for the engineered high affinity decoy receptor with 3 mutations shown in purple, again, there's a good on rate for the first 2 minutes, but now the off rate is markedly reduced. It just doesn't fall off that well. And this gives you excellent affinity in the picomolar range, comparable to the best monoclonal antibodies that have reached the clinic. That high affinity translates to excellent neutralization of authentic SARS-CoV-2 in tissue culture. In these neutralization experiments here, the wild type ACE2 decoy receptor is shown in gray. The engineered high affinity decoy with 3 mutations from deep mutagenesis is shown in purple. It works much better. And then it works even better if you convert it from a monomeric protein to a dimer by including a natural dimerization domain in ACE2. For free, we also got excellent neutralization of SARS-CoV-1, even though we never considered classical SARS in the engineering process. And this suggested to us that we probably had breadth against many other coronaviruses that can use ACE2 as an entry receptor. The question arises, did we choose the best 3 mutations to put into our engineered decoy receptor? We used human intuition. We've gone back using TL mutation from Diwakar Shukla's group. And TL mutation, the algorithm, it uses one of these popular POTS models, which includes the couplings, the covariation between different residues of ACE2 homologs. And it also includes the single site variation. So this information tells you something about what ACE2 sequences can give rise to a functional and folded protein. And then using a transfer learning algorithm, you can then import the deep mutagenesis data. And so using TL mutation here, this is looking at the binding data to the viral spike. The wild type protein is shown here with this dashed line, a couple of negative controls. These are a bunch of double mutants that they predicted with TL mutation should have good affinity. Some of them aren't much better than wild type, but some of them are quite good. And then these were our best single mutations that were combined into double mutations and into our favorite triple mutant variant here, which we're hoping to soon have in an IND program. And what you can see is that human intuition also does very well. There are a couple double mutants from TL mutation, which are comparable to our favorite triple mutant. However, for reasons that have to do with immunogenicity, we prefer our triple variant. So human intuition is good. TL mutation also pretty good, but this remains our favorite decoy receptor, which we are advancing. The SARS coronavirus, of course, is not static. It can mutate itself and change and adapt. If you look at the sequence variability that is observed amongst coronavirus strains that all use human ACE2, at least in tissue culture found in nature, then what you find is that the ACE2 binding surface right here of the receptor binding domain is actually, even though it's functionally conserved, it is permissive to sequence variability. And this raised the question, well, does our decoy receptor with 3 mutations in it, can it still bind to all of these variable coronaviruses? And I'm pleased to say that it does. It binds to these 5 bat coronaviruses and the 2 human SARS coronaviruses with low nanomolar or picomolar dissociation constants, much tighter than wild-type human ACE2. We have done a deep mutational scan on the receptor binding domain in the context of full trimeric spike protein expressed on the surface of a human cell, and we've looked for mutants in that library that can discriminate between wild-type ACE2 and our engineered decoy, and may therefore be the basis for resistance. The short answer is we don't find any such mutants, but what we did pull out were some mutations in the viral spike that massively increase affinity for ACE2, and in particular, the standout mutation was N501Y, increased affinity 20, monovalent affinity 20-fold less when you consider avidity on the right here. And of course, this N501Y mutation has since turned up in these highly transmissible lineages from the UK, Brazil, and South Africa, which goes to show how deep mutagenesis can really give you preemptive information on evolution of viruses out in nature. With that, I will finish. Many people contributed to this work. Here's my current lab, past lab members funding more collaborators than this, but these are the collaborators that contributed to what I showed, and in particular, working with a team at UI Chicago. They now have some simply spectacular data on in vivo efficacy of our engineered decoy receptor targeting authentic SARS-CoV-2 in an animal infection model. So that will be the next story coming out. And with that, I am happy to take any questions that people in the audience may have.